What I'm going to talk about is sort of, similar of what I spoke yesterday, but a little bit different in terms of what happened. Yeah. So I will talk about the leaf growth, and during the process of growth, how the surface curvature is maintained. Talking about curvature. So this, of course, is a linear curvature, and because it's a curvature in one dimension. And again, talking about dimension, what we know, we have learned in our school is there are 3 dimensions. This is 0 dimension, and the object, as an example that I have painted here, you can see. That is, you can't see, because a 0 dimension object you should not be able to see. So we all are taught that a dot or a point is a 0 dimension. So I can't really draw a dot with a 0 dimension. There will be some dimension when you actually put this one. There are 1 dimensions, either straight line or bent line. And according to definition, that it should have expanse only in this 1 dimension and not in the other dimension. You can't draw it. It's just imagination. 2 dimension, again, it should have expanse in 2 orthogonal directions or dimensions, but should not have any thickness. Again, show me one object, which is 2-dimensional. It's again a mathematical concept. So what we actually see in this world is the 3 dimension, right? That's the only dimension which exists for us, because there are some people who would say, no, there are 4 dimensions. There are more than 4 dimensions. But for a lesser being like me, there are only 4 dimensions that we know, and this is the dimension that the physical world has, or we perceive. And all these others are mathematical concepts. But with approximation, there are quite a few different types of organs which have 2 dimension. I'm saying it's approximation because they mostly grow in the XY plane and they hardly have any thickness. And that's the leaf that we have spoken yesterday, and he has spoken about petal and sepal and leaves and so on and so forth. Drosophila wing, the insect wing. Not that they do not have any thickness, but you can ignore it because that is fixed right from the inception of the organ. An Arabidopsis leaf roughly has 8 to 10 cell thickness, and that is determined very early on, and that does not change. So nothing is happening when the leaf is growing in terms of the z-axis. All the changes is happening in the planar axis. So you can consider that, approximate it as a planar organ. Consider this one. It's a huge leaf like a banana leaf. You can consider that as a thin sheet, right? Or a small leaf, doesn't matter. It's a thin sheet which actually has an enormous possibility in terms of making shape. But almost all the leaves make only one type of shape, shape in the sense, in terms of curvature, is all of them are flat. They do not really fold into the 3rd dimension to make more complex structures. Whether it's a small leaf or big leaf, and that makes sense because it's connected to the function. Unless it is flat and properly oriented toward the sunlight, it won't be optimum in terms of the sunlight. So there is a functional constraint in that, but how it is maintained is something which is not known. Yeah. There are variations, but roughly about 8 to 10 thickness. Even these 2, exactly. So I don't think it really depends on, but of course there are, so because it's big, it takes more, sort of stress, and that is taken by the vasculature, which is much tougher and many more in this one. So I think the weight is mostly borne by the vasculature, which is underlying the. So yesterday you saw about the section of some 3 different leaf types, and all of them have roughly, hibiscus and croton and Arabidopsis at 8 to 10 thickness. So as you saw that this of course are the mature leaves, and they are flat, but they are flat not only at maturity but throughout the developmental stages. But there is a problem here because we have seen that a small leaf does not grow uniformly, right? We have seen that when it matures, the growth is more from here, so there's a gradient of the growth. And if one region is growing at some rate and another region is growing at a different rate, then that actually can potentially cause some problem in terms of maintaining the flatness. And that you can, so the question is how the flatness of the leaf is maintained, even though the leaves are so big and growing from a very small area to a huge area. So that you can, I can explain with a cartoon. So this is a flat, suppose, consider this is a leaf which is flat and very small and is growing in the planar axis, and it can maintain the flatness as long as it is growing uniformly. So that is different regions are growing uniformly. But if you apply constraint in that uniformity, for example, if you apply constraint in growth in the periphery and not in the center, then center will grow more, and then it will lose its flatness, and finally it will become a cup-shaped structure. And we have in real life instances. This is not a flat anymore. You can have the other possibility. For example, you can apply constraint in the center, and then periphery continues to grow. In that case, it will buckle to form a saddle shape, or if you are hungry, this is probably a better example, the potato chips. This is not Pringles, by the way. In fact, this happens, the potato chips are, a saddle shape because they do indeed expand differentially, just like that. When you take a section of a potato, it is flat, and when you put it in the hot oil, it expands because of the water is escaping as steam. And when it is escaping, it is getting more surface area at the edge compared to the center. And then the edge expands at a faster rate than the central region, and that actually causes like this. So you have sort of three different possibilities. In principle, a growing surface can start with a flat and it can maintain as a flat surface, or it can have a cup-shaped structure, or it can have a saddle-shaped structure. But most of the leaves are flat. This can be explained mathematically, or geometrically rather, by Gaussian curvature. So again, a cartoon to explain that. So this is a hexagon, and this can be divided into 6 identical triangles. And you cut along this one and take one triangle out, and then again join them back. It will not have the flat structure anymore. So it will be like a cup-shaped structure. Right? So basically what's happening is the planar space is invading into the 3D. A 2D, apparently 2D space is invading into the 3D in one direction. Here it can be the other direction as well. So this is positive Gaussian curvature, surface curvature. If you do the other way, so what is happening here is you are removing more from the periphery than in the center. So that's why it is making a positive Gaussian curvature. If you do the opposite, you cut along this line and insert one extra, you are adding more surface in the periphery than in the center, and it will form a structure which is like a saddle-shaped structure, which is called negative Gaussian curvature. And this was actually shown experimentally by material engineers by an experiment about 10-15 years ago. 10 years ago. It's a nice experiment. So this is how it was designed. So what they did is they actually make a flat structure with a polymer. And the way they made is mix the two monomers. And if you mix them, within a minute they form, they polymerize and then form a structure which is flat. While mixing, so this is, suppose monomer A and this is monomer B, and only if you mix, it's just like, acrylamide and bisacrylamide, the derivatives of that. And if you mix them, while mixing, you can change the concentration. So there'll be a concentration gradient from the center towards the periphery, okay, such that there is more concentration at the center and less concentration at the periphery. If you heat this structure beyond certain critical temperature, it will start shrinking. But the extent of shrinking actually depends on the concentration. Lighter it is or more dilute it is, it will shrink more. So shrink is sort of negative growth, right? So there will be more shrinkage here and there will be less shrinkage here. Or in other words, if you take the opposite of that, there's more growth here and less growth here. So what you get is something like that. The actually experimental also you see exactly that. So this is what the experiment they did. So this is the concentration which is from the center towards the periphery is decreasing. You get a structure something like this. This is actually reversible. When you bring the temperature down to normal temperature, it goes back to the flat structure again. So it can keep on, happening, go back and forth. But if you have the opposite gradient, that is lower concentration at the center and then higher concentration at the periphery, then it makes a structure like this, which is sort of similar to negative Gaussian curvature or even higher order because it has waviness in the. And depending, by tweaking with the concentration, you can make a great deal of, structures. So these are the two positive Gaussian curvature. What they did is they measured the Gaussian curvature here, and this is the negative Gaussian curvature. And you can see that in the positive, so this is the flat structure, which is Gaussian curvature 0. And for the positive Gaussian curvature, there is a noise, but it is always higher. And here for waviness, it sort of fluctuates between positive and negative, positive and negative. And by changing the concentration, as I said, you can make all different kind of structures. And these structures are pretty similar to the structure that you see in nature as well, especially in leaves. So using this principle, what Schmick did, we call him Schmick because his name can't be pronounced even by the Europeans, so we call him Schmick and he's happy with it. His colleagues call him Schmick. So what Schmick did, he did modeling where to begin with, there is a flat structure, not this one, a flat square, and he added morphogen, which is blue colored here, in different regions, and then model and then see how the structure looks like. So if you put the blue color or the morphogen here in the center, you see that the center grows and the periphery doesn't grow, so it sort of makes a cup-shaped structure. If you make a gradient, more morphogen towards the periphery and then slowly decreasing to the center, it buckles into a negative Gaussian curvature. So one direction goes like that, the other direction goes like this. So the Gaussian curvature, a curvature, if it is a Gaussian curvature, negative or positive, you will be unable to flatten it even if you press it against a flat surface, right? Either you have to introduce cuts or it will fold over each other. That doesn't happen for the flat. And if you change the gradient, for example, it quickly gradient drops to 0, and then here there is no morphogen, then you see this wavy kind of structure that you see. So these kind of models actually is pretty similar to the structures that we see in nature. For example, this is one plant. The leaves are wavy. This is asplenium. And if you look at the structure of a single leaf and compare with the modeled one, it's amazingly similar. So simply by putting some morphogen, growth-inducing morphogens in the periphery in a gradient towards the center, you can make a very realistic structure which you see in nature. There are other examples as well. For example, these are complicated ones. So this is a cabbage. It's a kale actually, but it's a cousin of cabbage, one variety of cabbage, or lettuce for that matter. You see a lot of waviness towards the periphery. And again, by changing the concentration of the blue color, you can make, generate structures like that. You can, so in the model, he didn't use the positive Gaussian curvature, but in nature, you see the positive Gaussian curvature as well. For example, yesterday I showed you the hibiscus, which has flat and the zero Gaussian curvature, but there is a variant of it, which is the Hawaiian hibiscus, which has a positive Gaussian curvature. And this has something to do with, and as you see, so when the photograph was taken the day before it rained, so you can see the water getting collected here. New function, new shape, new function. And you don't need that for a leaf, for optimal sunlight, you don't need this one. So this is, of course, has less adaptive advantage than the flat one. But you do see variants where you have a positive Gaussian curvature, or the Victorian lily for that matter. This will also have a positive Gaussian curvature. So how to measure it? Because it's not very easy to measure Gaussian curvature, positive Gaussian curvature. So what you do is, in nature, you see, so these are variants, as I said, these are, very few species which are negative or positive Gaussian curvature, and majority of the species have. So what is so to study the genetic control of the flatness or the Gaussian curvature, you need to choose a model system where there are a certain number of genes, and mutate the genes one by one, as the geneticists do, molecular geneticists do, and then see whether by eliminating a gene function, you can generate negative or positive Gaussian curvature. So that is a principle of elimination or the mutation that that geneticists use. So this is what you need to do. So these are the two species that I show here. As I said, that almost all the species have zero Gaussian curvature. So this is the snapdragon, and this is Arabidopsis. You can completely flatten them. So just ignore the shape of the leaves and see only the flatness. You can totally flatten them, and they have zero Gaussian curvature. However, it's possible to generate by mutating some of the genes the structure which will make a form which is pretty similar to either negative Gaussian curvature. And you see there are waviness on the periphery. Presumably there are more growth in the periphery than in the center. We'll see that in the antirrhinum, or so only one gene is out of roughly 26,000 genes between this wild type and this one. Only one gene has been mutated, and other 25,990 gene is normal. So that gene is responsible for this one. If you mutate the similar gene in Arabidopsis also, you see this the similar structure. So these are not necessarily serrations. These are actually extra growth. So it is going in the z-axis and then forming this kind of waviness. Or if you mutate some other genes, for example, in Arabidopsis, whatever the name is, doesn't matter. One gene, if you mutate, you get a structure which is like a cup-shaped structure. Or the other one, which is p-pod, which is a very entirely different gene. Actually, two genes are mutated here. So it is possible by removing the gene function, you can change the curvature of the surface curvature of the leaf. But it's not easy to measure. I mean, this is, if you look at it, it looks like, negative Gaussian curvature, or this looks like positive Gaussian curvature, but to what extent it has changed. So for that, you can have some sort of, derive some empirical formula. For example, so this is a flat structure with R, and we all know that the perimeter and the area where it, is 2 pi r and pi r square. And since we know that the curvature is generated by differential growth or the matrix changes between the periphery and the center, you can take a ratio of the two. Okay. So for example, of course, this is R and this is R square, so you can take a curvature parameter which is root A, so that the R gets cancelled and all you have is a number. So this P by square root A will remain 3.6 if and only if this is a 2D structure. Okay. If it is deviates from the two-dimensional space, then it will be either more or less, and in fact it will be it's 3.6. The parameter is 3.6 if it is flat, and it will be less than 3.6 because there is more growth in the center than in the periphery. So P is and it will be more than 3.6 if it is. But this, of course, is for a circular structure. It may change depending on the shape as well, but mostly on the. So one mutant that I showed earlier is the mutant in Snapdragon called CINCINNATA, where you do not really see much difference between these two. This is a wild type, and this is the mutant where one gene has been mutated. The leaves are initiated. Their arrangement is pretty similar. The two leaves on 180-degree angle, and there is a 90-degree angle between these two and the successive leaf pair. So it generates like that, which is called phyllotaxy, or the arrangement of leaf on the stem. That is pretty similar. All you see is only the leaf structure is different. And so how different it is? So you take a mature leaf from here and compare. It sort of folds or becomes wavy towards the periphery. Presumably there is more growth towards the periphery than in the center, which will tell that it has overall negative Gaussian curvature. Of course, there are local curvatures which may be positive. How to measure that? It's not for this. It is easy because you can flatten it under two glass plate and it will be perfectly flat, and there won't be any folding over each other. So this has zero Gaussian curvature, but this probably not. So what you can do is you can of course it will have certain shape, and you can express the shape by a ratio of the length and the width, and you can do that same here, which is not probably very difficult because you just stretch it along this axis and then measure what is the length and what is the width. But curvature is not that easy. So what you can do is you can just cut it into pieces. Cut it into pieces small enough so that you can flatten them under 2 glass plates, and then you can measure very easily area as well as the perimeter. So you use ImageJ or whatever, MATLAB or whatever, and you can measure that. So you can have extract a lot of parameters, the shape parameters or morphometric parameters, the length and the width and the perimeter and the area, and then try to calculate the flatness parameter that I was talking about, which is p by square root a. So for wild type, you have certain length and the mature leaf I'm talking about, and you have certain width and the ratio is about 1.5. Whatever the numbers are, but for the mutant the ratio is 1, so it is a sort of more circular. So not only the curvature, it also changes the shape, and this is pretty similar to what a perfect circle would have. So if you do this, measure this at different developmental stages. So this is the leaf, wild type leaf grows in this axis. So initially there is not much change between the two. It is sort of long, and as the growth process continues, you see that it maintains its shape, but this fails to maintain its shape, and that's because it become more circular. This is a perfect circle probably, and this also is a circle. But later on, this has been made circular by pressing in the glass plate. It is more than circle. There is extra growth, which you can see that is folded over each other. So that actually is another measure of how much negative Gaussian curvature it has, because what you can do is you can take a crinkly leaf and force it between 2 glass plates and see how much is folding over each other, right? And if you see that under, against the light box, you can see, you can measure those area, and you can double that area because it's folding like this, right? So you can double that area here and then say that that is the extra area that it has got towards the periphery. And if you plot it as a function of the developmental growth, which is shown here. So this is the leaf is growing from very small to big, and this is the fraction extra that you have over the leaf area. For wild type, it will be totally 0, of course, because there is no curvature at all. It's 0 curvature. But initially, for the mutant, it stays 0, and then as you see that there is no folding over each other, whereas here it starts folding. You see that some fraction is extra towards the periphery. So that actually indicates that you have excess area which is going into the z-axis, which is indication of the negative Gaussian curvature. So if you actually measure them, the p by square root a, the wild type has some value, this has some value, but this p by square root is 3.8. Of course, it is deviates from 3.6 because the shape is elliptical and because it's dependent on the shape as well. But the mutant, which is circular as we have seen, is if you measure the p by square root, it is more than 3.6. For a perfect flat circle, it should be 3.6. And if you plot this value as a function of as the leaf grows, so this is about roughly 32% increase compared to this one, and that's enough to destroy the curvature. So what you're seeing here is the leaf is growing in this axis. This is the small leaf, and these are the bigger leaves. And the curvature parameter, or p by square root a, which is the blue is for the wild type, you see that it doesn't really change much. Initially, it changes a little bit. That is because the shape changes from the elongated shape. It becomes elliptical, but by and large, it remains similar. This leaf, which is circular, it starts like wild type and then reduces a little bit because of the shape change. And at this stage, it is completely circular and touches the value of 3.6, which is for a flat circle, and then later increases because of excess growth towards the periphery. So this has negative Gaussian curvature. So what has gone wrong? I mean, we know that the shape has changed. We know that the flatness is lost as the leaf grows. So one way of crude way of measuring the growth is we know that there is a growth gradient in the proximity distal axis. So you put some dots and then see at early leaf, and then see how the dots are the fate of the dot at the maturity, as we saw yesterday, and do the same thing for this one also. What you see is, so this is, highly, there is a gradient, and then you see that dots are collected towards the tip, whereas here it is more sort of homogeneous. So the maturity that moves from the tip to the base that we discussed yesterday is probably, either it has become more homogeneous or it has reduced or whatever that we can see, but it has been perturbed. So there is some growth defect in this axis. And what you saw yesterday is that maturity which is initiated at the tip first and then probably gets spreads towards the base also affects the cell size, right? Because cell size is a measurement of maturity. So cells are bigger here and cells are smaller here. If you take a small leaf like this is what it looks like. Okay, so the movement of the maturity is slowed down here. And if you take a growing wild-type leaf, you know that there's more mature, there's somewhere in between it is less mature and all. So this should be different in the mutant if you see that the movement of the maturity is changed in the mutant. So what you do is you measure the cell size here, here, and here in the developmental axis that is at different stages. So these are the small leaf and this is the big leaf in wild-type, and you see that the cells at the tip. This is the cells at the tip, matures very early on when the leaf is really 2-3 millimeter long, and then it increases and then goes to this value. The base remains, this is the log value, so there's a lot of things. So the base remains small for a long time and then it increases. At the end it is the same. So it starts with the similar size and it ends with the similar size, but as you saw yesterday that it takes sort of different kind of kinetics. And the mid cells is somewhere closer to the tip cells. If you do the same thing for the mutant, the tip cells, like the wild type, it matures early on. The basal cells remain young and proliferative for a long time and then it matures. But the middle cell actually behaves like a basal cell in the mutant rather than the tip cells in the wild type. So that says that the maturity that is moving down is slowed down, and the cells which is downstream to the maturity front is slowed down. So that's why the cells downstream remains proliferative for a longer time. And you can actually measure that by gene expression as well. For example, in this case, what you do is you take a young leaf and then cut into small pieces, and you do some biochemistry to see where the cell cycle genes are being expressed. In this case, histone 4, because histone 4 is specific to the S phase, and it indicates the proliferation. So if you arrange those, so this is about 10 micron sections. So obviously, if you have a 6 mm leaf, you have few hundred sections, but you arrange them from the tip to the base and extract the histone expression data, the red color. As you see that this is what the histone expression is. We have seen that there is a gradient. There's more histone here, and then the histone is cleared out because those are the mature regions. But in the mutant, even though it is sort of similar stage, you see the histone is there almost till the tip. So that's because the maturity is not really moving down as fast as it is moving in the wild type. But that doesn't really explain, even though the maturity of the movement of the maturity is reduced, that may explain why there is more number of cell or why it is bigger. And of course, this is bigger in size, but that doesn't really explain that why the flatness should change. So what we did is we looked at the cell division or the growth in the orthogonal axis. So the data that you have extracted from here, all these 620 sections, you use that data to make a cartoon. Wherever the color is there, this black color, that is and the intensity of the color tells you the intensity of the histone. Okay. So there's in wild type, there's a lot of histone, and then slowly it goes down, and there's no histone at all after now. And here, so there is a sort of transition zone here where on the top side, the cells are mature, and then bottom side cells are still proliferating. And of course, this is not a line because there is a gradient of it. And here that is reduced, so you can see histone towards the tip, and all right. But if you consider the orthogonal axis, what you see here is, for example, here. So of course, this section does not have any histone because it's mature. This has histone everywhere, so there is no difference in the medio-lateral axis. But here, you see more histone dots on this side than on the outer side. And so what it means that the maturity that sort of progressing down has mildly concave shape, and that is needed for keeping the flatness. that changes dramatically in the mutant. If you take a section here, you see there is a much more cell division or the histone signal here towards the peripheral region than in the central region. So if you measure the shape of the arrest wave, it is sort of more, almost like a round or highly convex. And that is can explain why it is losing its flatness, because there is more cell proliferation and more growth in this region than in this region. And this this transition zone actually is maintained throughout the leaf growth, even though it moves from the tip to the base. But as the leaf matures, it is constantly there on in the leaf. So it's actually affecting the entire, the leaf. This region must have seen the transition zone, a day earlier or something like that. So the change of this shape can explain why the leaf loses flatness, and why it is a negative Gaussian curvature, because there is excess growth in the periphery than in the center. And excess growth in the periphery will, excess growth in this axis will account for the wider leaf. This is a much wider than wild type, and excess growth in the proximal distal axis will actually account for the waviness of the negative Gaussian curvature that you see in the mutant leaf. So, I mean, you can put all this together and say that when the leaf is really, really small, since cell division is the default state, the whole leaf is proliferating and growing, and the tip stops growing first, and then that sort of arrest information comes down, but it comes down in this shape if CIN is not there. And the function of the CIN is to bring this down, not only to down and bring it down faster, but also change the shape of the front of the maturity array so that it becomes flat or mildly convex. And that explains. But how the CIN does it? One possibility is CIN itself is expressed in some sort of gradient manner in the medio-lateral axis, or there is some sort of gradient already there which CIN is responding to. So if you see the expression pattern of the CIN, so what you do is you take a section like this, transverse section, and see where the CIN is expressed. So this is one section of a leaf in the transition zone, and you can see so this is the RNA. So you can see there's more RNA towards the periphery than in the central region. So, so that means that, if you remove the CIN, there is more cell proliferation, leaf is bigger, so this is a negative regulator. And that negative regulation is more in the periphery, probably because the cells, the default state is that this region will proliferate more. So there is more CIN here to sort of counterbalance, and less CIN here, so that the growth is coordinated. The same thing happened for if you do an immunohistochemistry with the anti-CIN antibody, you see there is more protein here than with cells. So both the RNA and the protein is there more in the periphery than in the. So which means that in a growing leaf, which is actively growing and not and sort of has both cell proliferation and maturity, the tip is mature already, shown by the white color. The base is shown by green color, which is proliferating, and this is the transition zone where the cells are sort of exiting cell proliferation and entering into the maturity, and this is where the CIN is expressed. If we actually, so what we have here in this region, of course there is no gradient in this region, there is no gradient in this axis, but in this region there is more cell proliferation, and the cell proliferation and CIN has the opposite. There's more CIN here, and they reduce the cell proliferation. There's less CIN here, and more cell proliferation in the center to maintain the flatness. It's not very easy to see whether in the whole leaf. So this, of course, a data, a cartoon made from the data. But can you really see that the CIN is expressed in this kind of pattern in a growing leaf? Not in Anthorrhinum, but you can do that in Arabidopsis. And as I said, it's a similar gene that actually controls the flatness, surface flatness in Arabidopsis as well. And this is a leaf from a transgenic plant, which is expressing the CIN. Blue-colored CIN protein, or rather, Gus, which will make blue color, and the CIN protein fusion. And so this is the leaf, and you see that this is the mature region, and this is the proliferating region here, and the CIN is expressed sort of in this manner. And if you take a section here, which is the proliferating zone, there's hardly any cell proliferation here. You see there is more the Arabidopsis CIN rather, which is called GCP, and there is less in the so it sort of sitting there in this transition zone throughout the leaf growth, and the distribution in this axis is probably determines that how what the leaf curvature should be. So this is the sort of negative Gaussian curvature. If you can make somehow the this shape the opposite, then it's likely that you will generate a positive Gaussian curvature. It's not easy to do it because there are technical difficulties. There is no promoter which is expressed in a different way and all. But you can isolate a different kind of mutant where the curvature is opposite, as I shown you before. So this is we are seeing the top view of Arabidopsis leaf, Arabidopsis plant called rosette, and the mutant where one gene has been mutated, you see all the leaves are, not only bigger, but it forms a sort of positive Gaussian curvature. A mature leaf looks like this. This is the wild type, and you can see the bulges which is coming out. It's more growth in the center than in the periphery, so the bulges are makeup. And it happens pretty early on because these are leaf is growing, wild type leaf is growing in this axis. You can see it's 0.5 millimeter, really, really small. And at this stage itself, you have the curvature is quite apparent. And you can measure this curvature by, P by square root A, that curvature parameter that I described, in this axis, that is at different developmental stages, and then plot them. So this is how it looks. So this is the perimeter of mature, of course, and this is the area. 3.8 is the curvature parameter for mature wild type leaf, and in the mutant it is less. That 3.8 to 3.1 is roughly 18% change, but that's good enough to sort of show the curvature. It's a pretty sensitive parameter. And if you measure the P by square root A as a function of leaf growth, so this is the young leaf and this is the oldest leaf, and the black is the wild type, and as you see that initially there is some difference as you saw in the other species, that is snapdragon, because the shape changes very early on, but otherwise it remains pretty constant. But in the mutant, even though it starts like wild type, but it sort of deviates and then becomes less, and that is enough to introduce the curvature in the leaf. So you can a flat surface, you can make it a curve like cup-shaped by probably many different ways. One is there is excess growth with the cell proliferation or expansion in the center compared to the periphery. But it's also possible because there are multiple cell layers, right? So what you can do is you can expand the lower cells a little bit more, and then it will buckle like this. So there are two possibilities. What you did is we measured the cells at the top and at the bottom, that is the front side and the other side, and then just compare the two sizes and then see if there is any difference. Cells look like this if you measure them see them in scanning electron micrograph, and you can see in a mature wild type, these are the shape, cells, the pavement cells, the squiggly, the jigsaw shape that Olivier was talking about. And here, of course, the cells are small, and if you measure the total leaf area and the average cell size, you see roughly this is the average cell size, and this leaf is made up of around 15,000 cells, and this leaf is made up of 60,000 cells. So that is fine. But when you compare the cell size of the top side and the bottom side, you don't really see much difference. So the curvature is not probably because of the differential growth in the two different layers. So it's not in the z-axis difference. So the other possibility is probably there is a difference in the center and the periphery, as we described earlier. So what we do is we measure the cell proliferation by one of the histone, one of the cyclin expression pattern, and then see how they change along the time axis. This is the wild type, very young leaf, about 900 micron. And as you can see that there are a lot of cyclins here, and cyclin is clear from the tip because it's maturing. And as it matures, it gets restricted more and more, that you have seen yesterday also. If you do the same thing for the mutant, the first thing you see is even though it is 4 millimeter long, you see that there's more widespread cycling, that means more widespread cell proliferation. So the movement of the maturity that is, if you consider from the tip to the bottom, that has been slowed down, just like it has been slowed down in a mutant with negative Gaussian curvature in CINCINNATA. But what about in this axis? So what you do is you take in this transition zone and then divide it into, say, 4 squares and take a high resolution photograph from here, and you can extract the number of dots that is there in each square and count them and express them as a percentage of the total number of cells. Okay. So if you have 100 cells here, and if you have 5 cells are expressing, 5 dots are extracted, then it's 10%. So if you do that as in this axis, this is what you get. So this is the distance from the midrib. So this is the midrib, this is the midrib here, and this is the periphery of the leaf. So along this axis, if you come towards the medio-lateral axis, you see the Gus producing cells. Not necessarily it is dividing cells, because this is the Gus which is fused with the dead box. Of course, it is cycling, but still. So the Gus producing cell, if it is a marker of the cell proliferation, you see there's a mild gradient from the center to the periphery, and that is required for the wild type, because we know that is flat. And if you compare that with the mutant, what you see is that shape has changed along this axis. So there is more Gus, so this is pretty similar to at the center, like wild type, but then there is increases and then drops down. So there is excess cell proliferation in this sort of medial region, and that probably helps in causes excess growth, and the surface flatness is lost. But we know that so this is a positive Gaussian curvature. We know that we have another mutant like CINCINNATA. The CINCINNATA-like genes, if you mutate in Arabidopsis, you get the similar phenotype as I had shown before. So if you mutate that, then what is the shape of this the cell proliferation gradient along the medio-lateral axis? Is it opposite of what you see here? So this is the wild type, and this is the positive Gaussian curvature mutant I have shown earlier. And if you compare that with this mutant, which has negative Gaussian curvature, or the folds towards the periphery, the P changes from the wild type, A also changes, but what is important is the P by square root A, which is more than 3.8, which shows a negative Gaussian curvature in this mutant. And you can do the similar study. So this is the wild type, and this is the TNI mutant with the positive Gaussian curvature, and this is the negative Gaussian curvature of the cell proliferation. You measure in this axis and then plot it on this one. What you see is it's sort of opposite of the wild type. So the wild type, there is more Gus producing cells, and then progressively decreases towards the periphery, and here it increases. Okay. So so what it would mean is there will be more cell proliferation in the periphery than in the center in this mutant, and that will probably cause, hopefully can explain why the negative Gaussian curvature is there. And what will happen in the double? So if you mutate the TNI, which causes more cell proliferation in the center, and it's a positive Gaussian curvature. If you mutate this CINCINNATA-like gene, which is, doesn't matter what the name is, but in this Jordi means the CINCINNATA-like genes is mutated in Arabidopsis. If you mutate that, there is more growth in the periphery. And if you combine both the mutations in the double mutant, what will happen is there will be more in the center and more in the periphery as well. So that will be, leaf will be bigger, of course, but it may, bring back the flatness, but it depends on whether the relative contribution of those two genes. So if you make a double mutant, this is what it looks like, is much bigger than the individual leaves are much bigger than either of the mutants. And if you do the similar study, that is, morphometry, and then measure what is the perimeter, this is a single leaf of this double mutant. You measure the periphery and the center, and then take the ratio. It is much more than this one. So it, again, of course, it depends on the relative contribution of the gene which is functional in the center as opposed to the gene that is acting at the periphery. And if you measure the cell proliferation gradient along this axis, these are the two parents. This is what it looks like. And if you actually do the measurement, so this is the wild type, this is the Tharani, and this is the one, the positive Gaussian curvature, this is the one which is the negative Gaussian curvature. You see in the double mutant, it's a combination of both. You know, it's not like this gradient, but at a higher space. What you are seeing is there is more in the center and also more in the periphery. And as I said, it's probably because of the relative contribution that will tell how whether the flatness will be rescued or not, but it is not. So both the genes are, you know, functions independently and in two different domains. So what you can sort of conclude from here, that there is not only there is a growth gradient along this axis, there is a growth gradient in the medio-lateral axis as well. And there are some genes which are suppressing growth in the periphery. And if you mutate that, there will be more peripheral growth, and that will give rise to a negative Gaussian curvature. There are certain genes which are acting in the center, and if you mutate those genes, there will be more growth in the center, and then you will get a positive Gaussian curvature. You can explain this function by the expression pattern of this TCP, because TCP is expressed in this kind of gradient more in the periphery than in the center. But it's hard to explain how this gene acts more in the center than in the periphery, because it's expressed everywhere. So one possibility is, again, I mean, there is a gradient already there, and this gene is just responding to the gradient, which is not quite known. So I mean, this kind of study or the flatness can be done in homologous organs, homologous in terms of structure, in animal as well. For example, Drosophila. What I don't know is whether there are a lot of mutants in wing which is messed up, but I don't know whether any Drosophila wing which has a cup-shaped wing or not. But never mind, they are homologous structure, so you can compare them. But of course, I mean, the mechanism probably will be very different from the mechanism. So for example, this one, so in both the cases, so in this one, it's initiated pretty early on in the leaf. So what you can imagine is there is some sort of medio-lateral gradient of something, and TNI responds to that. So if you remove that, then there will be more growth in the center than in the periphery. Simply by the distribution of the gene by the promoter activity, just like it is there in the TNI, because the TNI is present more in the periphery than in the center. So when you combine the two, you can't reverse in this case. It's probably because you have to balance the growth in the center or the deregulation of the growth suppression that you have done in the center and the periphery. Probably you need to balance it. And so if it is not balanced, you won't get a flat structure. You will still have a difference between the center and the periphery. And in the double mutant, clearly you don't have that balance rescued or returned to equilibrium, because comparable, because you still have a lot of negative Gaussian curvature in the double mutant. Possibly you can, depending on the strength of the mutants, one combination probably will work where you have the flatness. I mean, there's more growth in the center as well as in the periphery, yet you have got back the flat structure.
Yeah. So leaves are either flat or ruffled, as you showed in nature, but they're also either simple or compound. And is there a correlation between curvature and compoundness?
So you can have a compound leaf, but completely flat. Okay? So, but whether compound or simple, that actually depends on the activity of the margin. So antirrandom, of course, has totally smooth margin, and but the majority of the leaves there is some sort of new growth cone or serration, and these behave exactly similar to the tip behaves. So what happens is this is Miltos's work and Patrick. So you have a the pin protein which actually pushes the auxin from the base to the tip. That has one sort of this direction very early on in the early leaf. And the CUC gene, which is a transcription factor, is expressed in sort of in localized way. And then the function of this is, once this is expressed, it actually reorients the pin. And then pin's job is to push the auxin, which is a morphogen which promotes growth. And so here the direction was like this, and here is reoriented. So you have a new growth cone which will grow. That's how the serrations are generated. So a smooth line, now you have, by this simple method, you can actually make a wavy line. And by repeating this, you can make a, so if this grows out more than the normal, then you have a sort of compound leaf. So if this is a leaf, and if you can change it like this, you call it a lobe leaf or serrated leaf or whatever. There's a midrib here. So if this touches the midrib, then you call it a compound leaf. By repeating this, you can make more complex leaves, and like tomato and others.
I guess what I wanted to say is that it seems that it's more difficult to keep a leaf flat if it's compound. But maybe it's not true. I don't know.
Maybe because now you have more leaflets, and you have to. I don't think it's really coordinated between the leaves. If the same thing that is keeping one leaflet flat is probably working, because the what is going on here in this axis is the same thing is happening here as well. So you just use the same method to keep it flat. I'm not sure that is more difficult for it.
Okay. If no more questions, let's thank the speaker.